Hello friends, today we are going to study restriction enzyme. Now what is restriction enzyme? Restriction enzyme is a bacterial enzyme that cuts the double-stranded DNA. Okay? Restriction enzyme recognize a particular restriction site on the double-stranded DNA and it cuts on that site only. Okay, so various restriction enzyme has their particular restriction site. Okay, um, now restriction enzyme can form two types of cuts: blunt end and staggered end. Blunt end has no overhangs, no sticky ends. Okay, no overhangs and no sticky ends. Staggered has sticky ends. That means they have either 5' overhang or the 3' overhangs. Okay, now what is blunt? Blunt cut means this double-stranded DNA restriction enzyme will cut at the same position in both the strands. It will recognize that particular site, suppose this is the site, and it will cut on the same position so that there is no overhangs. Okay, now staggered. What is staggered? Staggered cut means suppose this is double-stranded DNA and it will recognize this particular restriction site, but it will cut at the different position. Okay, now this has this and this. It will form such that there would be overhang, either 3' overhang or the 5' overhang, right? If this restriction enzyme cut here and here, then it will form such type of fragments. Okay, now this will have this type of, okay, these two are this would be suppose 5' to 3' 3' to 5', so this is 5' overhang. Okay, now in this way, if a restriction enzyme is giving a sticky end or the overhangs, 5' overhang or 3' overhang, then they are forming staggered ends. Okay, now we are going to study some new terms that we use in restriction enzyme. Okay, now as we know, first term would be star activity. Now what is star activity? As we know, restriction enzyme need a particular standard condition. So if we disturb that to work properly, okay, if we disturb that restriction enzyme, then the restriction enzyme will not recognize the similar restriction site that it can recognize in the normal condition. It will recognize the similar one but not the identical one. Okay, now suppose this is double-stranded DNA and a restriction enzyme can recognize this site, okay, in normal condition, but in non-standard condition it will recognize somewhat similar to this site. Okay, suppose GACT. Okay, it is similar but it's not the identical. So the restriction enzyme will recognize this site in the standard condition and it will recognize this in the non-standard condition. Means it can recognize the similar restriction site but not the identical one. Therefore, the sticky ends or the fragments that it will form would be different. Okay, this phenomena is known as star activity. Okay, second term, isoschizomers. Isocaudomers. Now what is isoschizomers? Isoschizomers are the enzymes that can recognize the same sequence but cut in the same location. Okay, then suppose there is one restriction enzyme 1 and there is another restriction enzyme 2. Okay, this both restriction enzyme recognize the same sequence, same restriction site, and it cuts at the same position. Okay, suppose GATC. Okay, this both will recognize this site and it will cut in the same position. Okay, such restriction enzymes are known as isoschizomers of each other. Okay, then neoschizomers. Okay, now what is neoschizomers? If the enzymes recognize the same sequence but cuts differently. Same sequence but cut differently. Okay, now I will explain this. Now suppose this is a double stranded DNA. Okay, now the restriction enzyme 1 and 2 both will recognize GATC. Okay, but one will cut here and the other second one will cut here. So they recognize the same site, but they cut at different position. Okay, such enzymes are known as neoschizomers of each other. Okay, now third is Isocaudomers. Now, what is Isocaudomers? Isocaudomers are the enzyme that recognize slightly different sequence but produces the same ends. Okay, means suppose a restriction enzyme is there which can recognize GATCC. Okay, and there is another enzyme that recognizes GATC. Okay, so if restriction enzyme 1 cuts here and restriction enzyme 2 suppose cuts here. Okay, so the sticky end that it will form is GATCC, and here also GATC. Means they are recognizing the different restriction site, but still they are forming the same sticky ends. Okay, such enzymes are known as isocaudomers of each other. Okay, so today we have learned a lot about this. So I will recall once what we have learned today. First we have done restriction enzyme. Then what is restriction enzyme? It's a bacterial enzyme that cuts the double-stranded DNA into fragments. They can form blunt and staggered end. Blunt end has no sticky end, no overhang. Staggered end has sticky ends or overhangs. Okay. Now then we have learned the new terms. Star activity. In standard condition, the restriction enzyme will recognize a particular restriction site and cut at a particular position, but in non-standard condition it will not recognize the identical restriction site but somewhat similar restriction site. Okay, and this phenomena is known as star activity. Okay, then we have learned what is isoschizomers. Isoschizomers are the restriction enzymes that can recognize the same restriction site as well as it cuts at the same position. Means it works identically. Okay, such restriction enzymes are known as isoschizomers. Okay, then we have learned about the neoschizomers. What is neoschizomers? Neoschizomers recognize the same sequence restriction enzyme recognize the same sequence but cut differently. Means they recognize, suppose here they recognize GATC, both enzyme recognize GATC but they cut at the different position. Okay, and isocaudomers are the enzymes that recognize slightly different sequences but form the same ends. Okay, so that's it for today. Keep learning, keep sharing your knowledge. Thank you so much. All the best.